Hello. Um, Let me talk about our poster. The title is here on the screen. So we explored the possibility of using machine learning with a variety of clinically and chemically relevant data to predict ligand interactions. Pharos is the user interface for the Knowledge Management Center for the Illuminating the Druggable Genome, or IDG, program funded by the NIH. Pharos provides a wide range of information on biologically and clinically important proteins. We developed a framework to predict binding affinities between kinases and possible ligands with the ultimate goal of identifying possible drugs. Model inputs are binary vectors that codify molecular structure and gene ontology annotations. These are available for all kinases in Pharos. The outputs represent binding affinities to sets of ligands. We identify clusters of ligands based on molecular similarity between ligands. Thus, the model works with affinities between individual kinases and ligand groups or clusters. The dimension of the affinity vectors on the output side is the number of clusters rather than that of ligands. We train a multi-layer neural net, or perceptron, or MLP, to predict the 5,275 dimensional ligand group affinity vectors from the 2,770 dimensional kinase structure gene ontology annotation vectors. Here's a schematic of the MLP model. The input and output vectors are both mapped to lower dimensional representations using PCA or similar bases in the span of the available kinase vectors. Coordinate vectors have approximately 400 components comparable to the rank of the sets of vectors. They are typically truncated to 100 to 200 on the input side, respectively 20 to up to 50 or 60 components on the output side. The neural net is trained on pairs of input-output vectors chosen from the 455 kinases documented in Pharos. Performance is evaluated on validation and test sets of kinases set aside. The key aspect of a predictive model in this context is its ability to identify ligands or ligand groups, in our case, that have a significantly high affinity to a given kinase. In the training process, we focus on comparing likely ligands as predicted by the model with known ligands for the test, but also for the trained set of kinases. The plot on the right shows examples of spectra with known and predicted affinity values for individual kinases, so the x-axis is the kinase index. Non-hits, that is ligands that, the x-axis is the ligand index, yes. Non-hits, that is ligands that are not known to interact with a given kinase, have exactly 0 recorded affinity. Hits have true affinities close to 1. The predicted affinities are noisy, but in most cases the distribution of affinities of hits and non-hits are different, with hits in a higher range. Compare those plots to the two histograms. Each of them represents hits and non-hits for one specific kinase. So ideally, the predictive affinities for the hits of a kinase will form a separate mode from the non-hits. This is not generally the case, but the hit and non-hit components generally fall into partially overlapping distributions. In this plot, the blob sizes indicate the number of known hits for the respective kinases. Colors indicate the train validation and test sets. And the bars, of course, are predicted ranges of hits and non-hits. The identification of hits from the predicted affinity values is a typical binary classification problem. The predicted hits and non-hits are defined by a threshold value applied to the predicted affinities. The area under the ROC curve is a measure of the accuracy of the classification. An area of 0.5 is equivalent to random selection, and 1.0 corresponds to perfect separation when the two ranges do not overlap and thus provide perfect identification. The ROC curve is not the only relevant measure. We also evaluated precision and recall measures, values as shown in the plot. In summary, the results for recent runs are quite promising. The precision, we get the mean ROC area by kinase around 90%. The precision around 4.7%, the recall around 65%. The precision is low because the total number of hits is significantly less than 5% overall, which is where we put the cutoff here. Thank you.